Deinococcus radiodurans are a small bacteria that have a naturally evolved resistance to damage caused by oxidative stress. This was discovered due to their ability to survive exposure to high amounts of radiation, several hundred times more than the average cell. Several proteins have been identified to correlate with this oxidative stress tolerance. We have chosen 4 of these proteins to take from Deinococcus radiodurans, a simple prokaryotic cell, and insert them into brewer's yeast, a more complicated eukaryotic cell that shares functional similarities with human cells. The newly developed CRISPR-Cas9 genome editing technology will make this transition relatively simple. Our goal is to see if the protective effects of the Deinococcus radiodurans protein will effectively transfer to the more complicated yeast cell.
As previously stated, Deinococcus radiodurans are bacteria with a naturally evolved tolerance to damage caused by oxidative stress. Oxidative stress is caused by reactive oxygen species, which are highly reactive molecules that can be generated by radiation exposure or naturally occurring processes such as oxidative phosphorylation. These species damage essential cellular macromolecules and lead to the disruption of normal cell processes. This damage is known as oxidative stress and is implicated in many major human diseases, such as cancer, cardiovascular disease, and neurodegenerative disorders.
The CRISPR-Cas9 genome editing technology will be the workhorse of our project. This newly developed technology allows for easily programmed and highly accurate targeting of genomic edits at multiple DNA locations. It works like a precision-guided DNA scissor, cutting DNA exactly where specified. When we provide a DNA copy of our desired protein, the cell uses it as a template to repair the break caused by CRISPR.
Our progress so far includes successfully selecting a target in the yeast genome and programming CRISPR-Cas9 to target that desired location. We have also successfully copied our 4 desired proteins from Deinococcus radiodurans and are in the process of building the DNA repair template for the use with CRISPR-Cas9 system to insert into the yeast genome. By going from a prokaryotic cell to a eukaryotic cell, there are many variables to consider due to the complexities of the eukaryotic cell. This step has required the most time and effort so far. Once this template is built, we can proceed with the transformation step and test the yeast for newly added resistance to reactive oxygen species.
We have high expectations for our project. By examining the effects of Deinococcus radiodurans protein expression in yeast cells, we will better understand how these specific proteins work to prevent damage from reactive oxygen species in Deinococcus radiodurans. Additionally, we will be examining the potency of these proteins in preventing oxidative damage in a eukaryotic setting, an early step potentially leading to the development of new treatments for Alzheimer's disease, cancer, and other neurodegenerative diseases.